Hello, everybody, and welcome to the Alzheimer's Drug Discovery Foundation September Town Hall. Mark Roithmer, CEO of the Alzheimer's Drug Discovery Foundation here, and of course, the one and the only Dr. Howard Fillett, our co-founder, along with the Lauder family and Chief Science Officer for the last 25 years since the beginning. So, Howard, we have a big group on today, and we got a lot to get into. So, are you ready?
We're all ready. Yep.
All right, we should go. So, Howard, what a summer, right? I like to think about it as summer to remember. I really do think for Alzheimer's science, we're going to look back years from now and say the summer of 23, wow, a lot happened there, or the first cracks in the wall, right? So we have the lecanemab, which is the ASI and the Biogen drug that got full FDA approval. And then, of course, we were all in Amsterdam and saw Eli Lilly's presentation on donanemab. And, like I like to say, there were 7,000 scientists in a dark room that their slide went up with their stats of does it work or does it not work? And the whole room just broke out into applause. And I guess really that's the point. After years and years and years of failure in the field, finally, it's starting to happen and the first cracks in the wall. And Howard, I spent so much time with you and know that, through your career, you go back to time Alzheimer's wasn't even being taught in med school. But you always had in your heart about how can we help families and how can we help patients? And you've done it as a physician and a scientist. And now the breakthroughs are happening. And now you even talk about harvest time. So, Howard, it's kind of good to be you right now. So, Howard, just, give everybody who's on today just a perspective of what this moment in time means to somebody who's been working at this for over 40 years.
Yeah, thanks, Mark. It's really true. When I went to medical school, I never heard the word Alzheimer's. And when I look back in my textbook, basically Alzheimer's was not in the textbook. And then in my clinical training, I took care of hundreds of older people and never even heard the word Alzheimer's or made the diagnosis or wrote the word in a chart. And it wasn't really till about 1980 that I became aware of the disease by visiting a nursing home and seeing all these bedridden people that were at the end stage of the disease and learning that this was a chronic, uniformly progressive, neurodegenerative disease that was fatal. Because of the loss of the human mind and all of its functions. And so as a neuroscientist, which I also was at that time, it became a very interesting and relevant, interesting from the scientific point of view, how we could have a disease of the human mind that was degenerative and rob us of what makes us human beings, basically. And from a clinical point of view, how can we care for these patients? And in the last 40 years, we've just come so far. It's just amazing. Both on the care side, we have so many programs now to improve care for dementia patients. And on the scientific side, of course, as you mentioned, Mark, over the summer, we really had a proof that there was a long debate about whether beta amyloid was a good target for the disease, and if we got rid of it in some way or another, that it would have clinical benefit. And I personally think that between ADDF, which obviously ran into some controversy, but presented data on the monoclonals in their clinical trials, the Eisai Biogen Leqembi or lecanemab, and the donanemab, which is about to be decided upon by the FDA in the coming months. I think we have 3 different programs from 3 different companies that show similar findings, which is that if you get rid of the beta amyloid from the brain, there seems to be a significant slowing, about 35% of cognitive dysfunction, and even a stronger benefit, especially from the Lilly trials, I would say, on slowing the rate of functional decline. And so
let me punch in there with you a little bit, because you bring up several really, really good points that I want to make sure we drill down on. we've got a big turnout today, and we've got donors, we've got VCs, we've got the NIH, we've got scientists from all around the world. And let's just for a second focus on the last two drugs, the lecanemab, because it got approved by the FDA, and the donanemab, which is, about to be approved, we believe about to be approved by the FDA. Just those two drugs, right? And for someone like myself, I'm not a scientist, but business person in this field. They're both targeting the amyloid. They're both done in intravenously, right? But so how do they compare, and also how do they contrast? How are they different?
Right, well, they attack a similar target, which is beta amyloid, but what's called the immunochemistry is different. So the monoclonal from Eisai Biogen attacks one end of the beta amyloid protein. The donanemab attacks the same end where it's modified by a technical chemical term called pyroglutamate, which presumably makes the donanemab a little more active in terms of removing plaque. But they both remove plaque in a somewhat similar way. The difference really is in, I think, the frequency with which the antibodies have to be infused. The Leqembi is infused every 2 weeks by IV infusion, and donanemab is once a month. And I think for elderly people with Alzheimer's disease and their loved ones who have to care for them and bring them into infusion centers, that could be a difference in terms of which drug people might choose. In terms of side effects, they have, I think, similar profiles. They both cause this ARIA-E, which is basically small pockets of edema in the brain, which we think is due to inflammation from the immune reaction that occurs between the antibody and the plaques, and ARIA-H, which is microhemorrhages, which also may be due similarly to some immune reaction, but we don't really know. But there are appropriate use guidelines that have been published that I think Medicare, managed care plans, and other Medicare and commercial plans will adopt that kind of lay out the way these drugs should be properly managed, both in terms of safety monitoring, which includes MRIs of the brain about 4 or 5 times in the first 3 months of treatment to make sure that these side effects don't happen. The side effects on MRI that I spoke about may result in headache, confusion, dizziness, but most of the time when these occur in 20 to 30% of patients, they're not serious. About 1% of the time, I would say, they can be serious when there's a lot of edema, but that's very uncommon. And I think their benefits are somewhat similar.
Harold, let me go on that, on the benefits. So first of all, thank you. That differs them, really well, and also compares them really well. And the point that you're making is, from your view, they are safe. And if I'm putting words in your mouth, you'll come back to that. The other thing is, again, in not being a scientist, I hear this all the time about the two drugs, that generally they're about 30 to 35% effective. And I often get questions about that, 30 to 35% effective. Does that mean that 30 to 35% of people taking these, like, they're treated and everything's fine for the rest of their lives? Or does this mean it pushes out or delays? What does that 30 to 35% effective mean?
Well, first of all, it depends on which outcome you're looking at. Is it cognition? Is it function? Is it composite scale of all of these things? But I think for the purposes of, let's say, traditional FDA approval and the way we traditionally talk about these drugs, it's a 35% or 30%, let's say 30% slowing of the rate of decline over a period of 12 to 18 months. And with open label extensions, it would seem that these differences widen out, which is what you'd expect. So the improvement. But I think recent ways of analyzing the data have more clinical meaning. The donanemab drug was recently reported that 50% of the people who took donanemab during the course of the trial were stable for 1 year, while only 29% of the people who got the placebo were stable for 1 year. And I think reanalyzing the data in this way is really important, because I can say to my patients, as you said, Mark, this is going to slow you down, let's say 30%. But the fact is that when patients come into the office, we don't know what their baseline rate of decline is. And so it's impossible for any one clinician to tell any one patient that there is 30% slowing, let's say after 12 months, because we don't know what their baseline is. But the fact that people stabilize in a disease that's uniformly progressive, I think, is a meaningful outcome for families. If they came in after 6 months or 1 year on the drug and they're still stable, let's say on a very simple cognitive assessment or in the opinion of the carers, 1 year, that's a lot of time saved with saved cognition and function. And I think that's one way to look at the data that makes it clinically meaningful to patients. And that's, I think that's really important.
And Howard asked and answered. Good, excellent. I'm going to want to pivot, and as I'm pivoting here, I want just for all the folks who are on, I meant to say this up top. Howard and I are going to, in full, talk for about 35 minutes, and then we're going to leave a good chunk of the back part of the half hour of the end of this for Q&A. So you can put Q&A into the queue, and we have folks monitoring that, and they'll be feeding the questions into Howard and myself. So just on the pivot, I wonder if we could pull up there's an article and there's a quote from you from the Financial Times. And a couple of things here that I just want to say, and I go back to something you said, Howard, when you were saying about, there was a big debate. Is it amyloid? Is it not amyloid? And I know through all the years that I've worked with you, you've always said, there's no religion on this, folks. It's just science. And yes, amyloid has something to do with it, but it's not the be-all, end-all. And amyloid is a protein, and it's part of the biology. And I think I got into, I think several of us in the office got here into a saying about, yes, it's the biology, stupid, is how we talk in the office about it. But this quote, and Howard, I want to give you some additional kudos here. First of all, so great to see how much attention Alzheimer's is getting these days, number one. But number two, I recently heard from some folks from the outside, and they did an audit of who's being quoted in all these science stories. And I was proud to see, Howard, that at 30%, you have the greatest single share of voice outside of the drug companies themselves whose drug it is. So, but this was one that was particularly good, came out of the Financial Times, and folks, it's been up there and folks can read it. But what it really says is, hey, this is a watershed moment, but this is just the beginning. And that we're really going to need this biology of aging, combined therapies to kind of stop this in its tracks. So we talk a lot in the office, it's about the biology of aging, but Howard, could you just, touch on what is the biology of aging about? And then we're going to drill through a couple of particulars about it, but just overall, it's something that you've pursued your whole career. So if, give us a 101 version, Howard.
Well, there are pathways to aging that affect the development of multiple diseases of old age, diabetes, hypertension, cancer, Alzheimer's disease, other neurodegenerative diseases like Parkinson's, strokes. So there are fundamental mechanisms that really relate to how these diseases develop. And if you look at aging itself and the studies of aging through biogerontology, these 7 or 8 or 9 common pathways of inflammation, epigenetics, something called autophagy or proteostasis, mitochondrial problems with oxidative phosphorylation. And I know I'm using technical terms here, so I apologize if anybody isn't understanding exactly what I'm saying, but I just want to give some technical background to the common themes in research on aging itself really can be extrapolated to mechanisms of aging of the brain and Alzheimer's disease, metabolic disorders being another one with insulin resistance and problems with what's called the integrated stress response, and even senescent cells that occur in every organ that are inflammatory. And won't die. So running through that technical compendium of things that we know are typical of aging pathways that also are abnormal in Alzheimer's disease. And I think if we provide a framework, because aging is by far the leading risk factor for Alzheimer's disease. So it makes sense to go back to the gerontology research, which has been going on for well over 100 years, and learn from all of that, which has been published many, many times. The Journal of Gerontology was established in 1948, long before Alzheimer's came to the front of research in the 1980s, 40 years later. So I think there's a way to think about this that relates to aging as the leading risk factor and understanding the mechanisms, and then addressing each of those mechanisms with new drugs. And actually, amyloid deposition, the plaques have many different proteins in them, and we're just starting to recognize that the plaques are deposits in the extracellular space of the brain of many what are called misfolded proteins. And the reason this is important is because proteins that are made by the DNA in the cell and the RNA, they have a linear sequence, but they have to fold into a three-dimensional state that makes them functional. And when they get damaged by, let's say, oxidation from, just cellular function, then they tend to misfold and they become damaged. And the example I like to give on this is if anybody's had a cup of coffee and you throw some vinegar in there for whatever reason, the milk curdles. And what's happening in that case is that the milk proteins are misfolding. And that's happening in every cell, in every organ in our body every day. So through evolution, the body has had to make compensatory mechanisms to get rid of these toxic misfolded proteins. And I think this is a very important area because amyloid in its deposition is a toxic misfolded protein. And so is tau, which is the other one that we recognize typical of the disease. But we now know that the aging brain also has misfolded alpha-synuclein. That's something like 35-40% of people with Alzheimer's disease also have these other deposits that are called Lewy bodies that have a misfolded protein called alpha-synuclein. There's another misfolded protein that's been recognized called TDP-43 that occurs in Alzheimer's patients. And so we're beginning to recognize that there are many misfolded proteins that are dysfunctional and toxic. And my point is that, yes, we can get rid of the beta amyloid from the brain, but that doesn't result in 100% slowing. And one of the ways that we can maybe get there is to have a more parsimonious approach to these misfolded proteins by addressing this mechanism, which I described to everybody, called autophagy or proteostasis. And so we're just starting to invest in, because it's a difficult mechanism to create drugs. We understand more about the biology today of this mechanism, but we haven't been able to drug it because it's the difference between understanding the research on the biology, the basic research on the biology, and then the very difficult phase of translating that research into new drugs. So we're currently, and we have been investing in ways to help the body cells, and particularly in the brain, to get rid of these misfolded proteins. So autophagy is an important new role. Inflammation, and I'll just give you one more example, is
Howard, let me do this. Let me get in because we'll hit the inflammation side. So again, I think what we've heard and what we've believed at the ADDF for years, the brain's complex. It's not going to be one size fits all. It's not just the amyloid. So I want to walk through how we kind of get there. Well, what gets us from the 30% effective to closer to the 100% effective? And I want to start with kind of the most straightforward or maybe simplest of the concepts, and that's prevention. And Howard, I'm going to go through 2 or 3 different parts of prevention, but let's just start it with lifestyle. Howard, from your point of view, okay, what are the 3 or 4 key lifestyle things we know you can do that's going to push out cognitive decline?
Yeah, so you're looking at a picture, obviously, of Mia Kipapelto, who's our partner now.
We'll get to Mia, but start with just the 3 or 4 lifestyles.
Okay, well, you have exercise, diet, managing cardiovascular diseases, and including diabetes and hypertension, vascular comorbidities, and stress and isolation, I would say, are the five main ones that I think we can, but that Mia has actually pioneered in her studies in terms of prevention.
So let's get great, really straightforward on the lifestyle. Let's get into Mia a little bit from two levels. One, talk about the worldwide fingers that she's done that proves these lifestyle interventions. But then, Howard, just as importantly, move it into, because there's lifestyle interventions, but now we're starting to combine lifestyle interventions with treatments. So kind of think about combined therapies, prevention with the treatment. So Mia has done this finger study. Just give a quick on, why this is all fact, and then pivot us into what Mia calls fingers 2.0, and that's with the metformin. So take it away.
Mia's innovation was that prior to her work, most of what we knew about prevention was from what we call epidemiological studies or observational studies. So we would follow people over many years and see what was associated with a decreased risk for Alzheimer's disease. The problem with, while there's evidence, this is really what we call hypothesis generating. And the problem with that kind of study as a level of evidence is that it can only create associations between a risk factor and the development of disease like Alzheimer's disease. So the highest level of evidence is the randomized clinical trial. And no one had really ever run a randomized clinical trial of prevention of these risk factors to prove that they actually work, just like you'd want to do a randomized clinical trial with drugs. And so long and short is that I think the innovation that Mia brought is two things. One is she ran a randomized trial, but it was really one of an implementation science. In other words, we can tell people, manage your diabetes, manage your hypertension, don't smoke, don't drink alcohol, at least not in excess, and so on, exercise and eat a healthy diet. But people aren't necessarily compliant. It's hard for people to do. So what she did was she had a control group where people were briefly educated in 1 or 2 sessions on what they should do, and then left to their own devices, basically. And of course, many of the people were not compliant. And then in the intervention group, had monthly visits, basically, to make sure that people were being compliant over the course of the trial. And she showed that in people who were compliant, there was a decline in the rate of decline of their cognition with aging, and some effect on the disease. So this study was so highly appreciated that it's now being replicated in 60 countries around the world, where each country wants to see if in their population these kind of interventions work. What we're working with Mia on right now is sort of analogous to what went on with heart disease, where early on lifestyle risk factors and comorbidities like diabetes and hypertension were recognized, but that was as far as we got. But once cholesterol was recognized as a risk factor and a biomarker for the disease, then we could develop statins for the treatment of people that had the disease, that already had a heart attack, to prevent a second heart attack. And then ultimately for prevention, where people who didn't have a heart attack but had a high cholesterol, had high risk factors, could get a statin to prevent that first heart attack. And then what we're doing now is combining Mia's lifestyle program and comorbidity management with the leading anti-aging drug out there, which has a lot of interest right now, called metformin. So it's kind of like the finger study 2.0, where Mia will do the randomized trial with her various lifestyle interventions and comorbidity management, and add metformin to see if there's added benefit. And this, I think, will be the first of many studies where this kind of prevention model will be utilized to study how we can really use drugs that might have an impact on the course of the disease and preventing it. And of course, the other thing that's really in this new era is that this can be done for Alzheimer's disease. And the reason is because we have these brain scans that detect amyloid in the brain 20 years before people start having symptoms. And so we can, using blood biomarkers of beta amyloid ratios and which correlate with the brain, or even brain scans. I think it's going to be a lot cheaper and more practical to use the blood test. We can actually start to detect people before they have symptoms and then put them in the lifestyle comorbidity management and even drug treatment. There have been studies of the monoclonal antibodies for the prevention of Alzheimer's. Maybe we need to combine lifestyle interventions with monoclonal antibodies to see. So it opens up a whole new world of prevention that's made possible today because we have the biomarkers and we understand what the risk factors are.
Yeah, I was struck by when we were in Amsterdam and for Lilly's big announcement, and Mark Minton, who, that's his drug at Lilly, his ending comment was, if you take nothing else away from this presentation, is you want to know early because there are things you can do about it. And, Mia talks about, we're going to move off prevention now, but Mia talks about precision prevention. And we're entering a new partnership with the Fingers Brain Health Institute and the Alzheimer's Drug Discovery Foundation. Perhaps that's something we can talk about a little bit more deeply the next time we're all together. But it's really, just remember those words, precision prevention, because that's what this opens us up to. So Howard, it's interesting. You talked about the prevention. You talked about the amyloid drug with prevention. Let's talk about the combined therapies now that we have a standard of care that's coming out with this amyloid drug, but we know it's only 30 to 35% effective. And we kind of want to follow what cancer's doing, right? To get it to be closer to 50, 60, 70, 85, 90% effective. Talk a little bit about, combined therapies and what, as you look at it, what might we be combining these anti-amyloid drugs with?
Yeah, so just to start off the conversation with this slide here, you can see that 75% of the targets that are now in clinical trials are non-amyloid targets. And this is really quite different than the field was even 5 or 10 years ago, where 75% of the drugs in development were basically anti-amyloid or anti-tau drugs. So you can see when I mentioned the 8 or 9 different pathways that are involved in aging that are translatable to Alzheimer's, you can see basically the pathways that are being investigated now across this field that are non-amyloid pathways. The question is, as you mentioned, Mark, is how do we design new trials if these monoclonal antibodies, which are quite expensive and do have risks, there's no doubt about it. I think the risks, as I mentioned, are manageable, but there are risks. And so will we have to, as we go into these new pathways, do combination studies as evolved in cancer, where we would have, let's say, anti-inflammatories for the brain, but the trials will have to be done on a background of a cholinesterase inhibitor and a monoclonal antibody to amyloid to see what incremental benefit we get. And that would have to be driven, as you mentioned, in terms of precision medicine by biomarkers. And we are getting biomarkers for many of these areas now. So we'll be able to identify, let's say, people that have amyloid in their brain, they have Alzheimer's disease, but they also have evidence of neuroinflammation from biomarkers like GFAP or YKL-40, technical terms, to identify new biomarkers that reflect inflammation in the brain. And I think as we think about it, these trials are going to be more complex, perhaps more burdensome in terms of the requirement for the IV infusion, and more complicated, might even need more patients, because we're going to need 4 arms, at least the FDA, in conversations that some of our investigators have had with the FDA. They might want to see one arm which is kind of placebo, one arm which has a combination of the standard of care, and then one arm with combination of standard of care plus the new drug. So these trials are going to be more complicated. And what we don't have right now is kind of a consensus about what the design of these trials is going to look like. So I think that's the next challenge. One of the great advances that I believe we've had in the last 5 or 10 years is our ability in the field to really do excellent, rigorous, efficient clinical trials with biomarkers that are validated, but we don't necessarily have applied that that technical expertise in designing these trials and creating the statistical models that are necessary to test these new drugs on top of the standard of care. Now, of course, it is possible to run them just by themselves, but that wouldn't be the optimal way of going. And what we want to do is go from 30% to maybe 50% slowing of the rate of decline by adding on an anti-inflammatory. And I think combination therapies will have to be the way to go to prove that these combinations work. And that's really how cancer chemotherapy evolved in combination therapies as well.
Yeah, and Howard, I give you and the scientific staff here such tremendous credit. And it's not just that this is a theory that you've been putting forth that now is coming to fruition, but also that, you and the scientists talked about this in the office, and how do we come up with, how to do the best combined therapy trials. And you've put together an ad board that has representation of all the major pharmas coming. You are convening all the major pharmas, statisticians, people from the regulatory side, folks that have done this in the cancer years to literally map that out and put out how combined therapy trials should be done for Alzheimer's. So kudos to you and kudos to the ADF there. We're just going to go one last topic, Howard, here, and then we got a bunch of questions, so we'll open it up for questions. But that last topic, and you really touch on it about with the combined therapies. Combined therapies, you need precision medicine. That means you need biomarkers. And I was struck by, again, when we were in Amsterdam after the Lilly news, we have this great partnership with Gates Ventures and others around our diagnostic accelerator, right? And it's looking at things like blood tests and eye tests and digital and AI, how we can diagnose. And I was so struck by, we were meeting with several of the companies that we were funding in Amsterdam after the Lilly news, and they all came in and they said, geez, given the Lilly news, what's the landscape for diagnostics? And I think, easy for me to say, but the business term is it's a land grab. What can get to market now? Because we have gone from what has been a societal issue of how do you deal with Alzheimer's? Well, now a standard of care is coming. If a standard of care is coming, well, how do I know I want to know? Because now I can do something about it. So Howard, and we've been working with our great friends from Gates Ventures on this for a couple of years, and also the origins of ADDF have always been around biomarkers and diagnostics, along with preventions and treatment. So Howard, walk us through, like, you were the guy that, helped bring the first PET scan to market, and that's why these drugs are starting to break through. Got that. 2 years ago, the first blood test with C2N. So give us a feel of where we are on these diagnostics, the blood tests, and also the digital.
So it's really amazing because prior to the current era, there were no blood tests really for any neurodegenerative disease, Parkinson's, ALS. And so patients, generally speaking, to get a definitive diagnosis had to have a spinal tap or, in the case of Alzheimer's, a brain scan. And now we have a blood test. And I really think that's going to change clinical care because this blood test from C2N, which is a company we made an investment in a number of years ago, but other companies that are coming to market, like Roche, for example, is bringing a test to market. But imagine that the world has changed. There are now really kind of validated blood tests for Alzheimer's disease. A patient can go into their primary care doctor and they have a memory complaint, and hopefully they'll get some kind of brief cognitive assessment that's validated, like the Montreal assessment or the mini-mental that a lot of us use. And if there's a problem and the patient wants to know, or the patient and doctor want to know what the diagnosis is, which they'd want to know for any other disease, they can order a blood test. In 49 states, New York is the one state that hasn't been, the C2N test hasn't been approved, and they just came out with an even better test. So, once the test is done, within a few days, the blood test comes back, and we would know with a fairly reasonable degree of certainty, maybe 85%, that the patient has or has not Alzheimer's disease. And I think that really is important in terms of confirming that a person with the very early stages of cognitive impairment has something going on that potentially now is treatable because we have these monoclonal antibodies. So if they're amyloid positive, they can go to treatment. And also it increases the cohort of people in the community that can participate in clinical trials. And the big advantage in clinical trials with the blood test is the cost and the accessibility of screening for people to enter into these trials with a definitive diagnosis of Alzheimer's disease based on a blood test and perhaps confirmed by a PET scan, although I don't personally think that it will necessarily be required. And so this is really going to change because a big part of the cost of clinical trials is finding the patients and validating that they actually have the target of interest. For example, in the case of beta amyloid deposition, even either in the brain or on a blood test. So these blood tests are also what we're developing at ADDF with the Diagnostics Accelerator in partnership with Gates Ventures. Is if you recall on the previous slide, we had multiple other areas of interest that are being investigated now, and we need biomarkers for each one of them. We have biomarkers in development now that I think will come to market for detecting inflammation in the blood that reflects inflammation in the brain. This is really important for the next generation of anti-inflammatories for Alzheimer's disease. And there are other biomarkers being developed for the other pathways that we talked about several times during the course of this. So again, we mentioned precision medicine. We have to build this field on the back of biomarkers and the back of new drugs where the biomarkers are responsive to those drugs so that we know that the drugs are working. And basically, it's a new era. It's very exciting. And we're building this new era of treatments so that we'll have combination therapy. And I hope ultimately get to prevention of symptoms in the first place, but in people that express symptoms, be able to slow down the course of the illness to stabilize it for a long time so that people can retain their cognition and keep remembering their grandchildren till late in life.
Amen, brother. Amen. Okay, so Howard, we're going to get to some questions. You're going to love this first question that came in because it goes exactly to a point that you were trying to bring up before, which I so rudely steered you in another direction on, which is good. What role does inflammation play in Alzheimer's? And you've been alluding to it. I think we're good there. But the second part of this really is, what are some examples of promising anti-inflammatory drugs that are in the pipeline right now?
Right. That's a great question. Inflammation is complicated because there's systemic inflammation and there's brain inflammation. And I think all of us have the experience of when we have systemic inflammation from a cold, we express what's called sickness behavior. We want to go to bed, we feel a little depressed, we're tired. And some of these symptoms are cognitive. We're not thinking as clearly. And that's because there's a direct effect of systemic inflammation on the brain to prime cells, to prime the inflammatory cells in the brain called microglia to become activated. And when microglia become activated, they can do one of two things. They can be helpful to remove proteins like beta amyloid, or they can be harmful by expressing too much of an inflammatory response, which is damaging to cells. So inflammation is a hallmark of aging. It's very common with old age, but there's also a very specific type of inflammation that's specific to these microglia cells, which are the very specific resident inflammatory cells in the brain. And so many years ago, back in the 80s, I was involved in a study of prednisone for Alzheimer's, because that's how far back this inflammation hypothesis goes. And of course, that was a small study, didn't show much. I would think maybe we should even repeat it today to see if prednisone is useful for Alzheimer's disease. The reason I think that inflammation is so important clinically in the brain is that studies have shown, particularly Clive Holmes from the University of Southampton, has shown that people that have evidence of inflammation with Alzheimer's disease progress more rapidly than people who don't. And my lab back in the 90s showed that there was evidence of this increased inflammation in the brains of people with inflammation. So there were biomarkers for this going back even way back then. So the point was that he treated these patients with an anti-inflammatory drug, which is very common and commonly used in rheumatoid arthritis, called Enbrel or Humira, which blocks alpha, a cytokine, an inflammatory molecule called TNF alpha. And he showed that by blocking that, you could block the progression of the disease. So it seems like we know there are people that have beta amyloid in their brain and they don't progress. And it looks like those people might be people that aren't mounting an inflammatory response to the beta amyloid. And the people who have it, have beta amyloid and have the inflammation, tend to progress more rapidly. And some of the companies that we've been investing in, one is called Neurokine, another is Vaccinex, there is Immune, fourth is Neurotherapia. And I won't go into all the different mechanisms and approaches that these companies are using, but there's a lot of different ways that we can try to halt the neuroinflammation, which is very specific to the brain, and slow the course of the disease down.
All right, and the questions are just piling in here, so I have a cherry pick of just fabulous questions here. So, Howard, vascular disease and Alzheimer's, what work's being done?
Well, we know that 50% of people in the community that have dementia have who have Alzheimer's disease also have what we call a mixed dementia, which means they have vascular disease along with Alzheimer's disease. And of course, cerebrovascular and cardiovascular disease are highly prevalent in the general community and very common in patients with Alzheimer's disease. So it's very important, and Mia's finger studies show this, and there have been other studies, particularly looking at the vascular risk factors of hypertension, because that causes in the brain, it's associated with what's called not just large strokes, which we typically think of, but impairments in the microvasculature of the brain, in the deep white matter, where all the connections are between the various components of the brain that are functional and cognition and so on. And so we kind of have a biomarker now, the NINDS, the National Institute of Neurodegenerative Diseases, neurological diseases, is trying to build this field of looking at vascular dementia, specifically developing biomarkers for vascular dementia, because we really need those to look at these people with mixed dementia. Probably somewhere between 15 and 20% of patients in the community have pure vascular dementia by strict research criteria, where there's no evidence of amyloid, and the main cause, the primary cause of the dementia is vascular disease. So that's another subset. But the more common type of scenario is people that have both diseases, both a vascular component. And it's also true that the amyloid itself in Alzheimer's disease deposits in the blood vessels. And we're funding, we've invested in a very interesting company, I think, called Therini, which we started funding at the Rockefeller University in the academic setting, because it turns out that an important coagulant molecule in the blood called fibrinogen is processed to a smaller entity called fibrin, which helps clotting in the blood vessels when there's blood vessel damage. And what Katerina Segal showed at Rockefeller is that these fibrin deposits that are activated from platelets and so on in the blood to try to do coagulation, when there's amyloid there, they form very tightly bound deposits that damage the blood vessels, lead to blood-brain barrier leakage, and cause an initis of inflammation. And potentially even that's where some of these plaques start. And so Therini translated that. They'd licensed and got some of this information, and they're approaching clinical trials now with their monoclonal antibody to a very specific part of the fibrin molecule that blocks this interaction with beta amyloid in the blood vessel, but doesn't impair the coagulation that needs to occur when there's damage. And I think this is really a very innovative and novel approach to the vascular. And the last thing I'll say about it is it kind of connects. We don't know why diabetes and hypertension are risk factors for Alzheimer's disease, but we know that they really are. And it's probably at this what's called the neurovascular unit that this connection between diabetes, hypertension, and maybe even atherosclerosis occurs, to open up the blood-brain barrier, have amyloid deposits begin the process at that point of Alzheimer's disease as we know it. So,
So, fascinating, Howard, right? You've got combined therapies, the anti-amyloids inflation, the anti-amyloids vascular. I'm going to switch this. There's a ton of questions, but there's a question bringing us back to diagnostics and biomarkers. And the question is, what's going on in the digital world?
Yeah, great. I'm glad we brought that up. So clinical trials in Alzheimer's disease are really expensive. Like phase 3 studies are $300-400 million each. They take about 18 months. And it takes about half of that money is spent on just trying to find the patients. And generally, about 1,000 patients have to be screened to find 50 to 100 that really qualify for the trial. And so right now, this screening process is very expensive because if we use neuropsychology, it can be time consuming. And if we use the brain scans, it can be time consuming. So how do we reduce the cost of trying to find these patients? Well, we mentioned the blood test as one way, but on the neuropsych, which is important to identify people that actually have, let's say, mild cognitive impairment, generally it's done by a neuropsychologist, and it often takes hours of testing to find these patients and characterize them cognitively. But now what we can do is we can take the smartphone and have validated digital tests on the smartphone that test cognition in very sophisticated ways, in fact, in ways that are more sensitive than a neuropsychologist, because the smartphone has computer capabilities to pick up things like slight abnormalities in speech, which we've all experienced as we speak to older people or see them on TV and they have trouble finding words and, this kind of thing. We won't talk about the politicians, but we talk about that. Well, now what we're developing through something called the Speech Consortium, again in partnership with the Gates Ventures through a diagnostics accelerator, is going to be a very large database of about 3,000 people de-identified who have actually at various stages of normal, mild cognitive impairment, maybe mild dementia. Their speech is going to be analyzed in very sophisticated ways. And those speech abnormalities that we pick up, which are going to be very sensitive, will be put into a database at the ADDI, which is managed by, it's the Alzheimer's Disease Data Initiative managed by Gates Ventures. And that information will be available to the general research community to develop algorithms so that people can be screened in the community on the iPhone. But the other thing that this does is every time a patient goes into a clinical trial, let's say they're visiting the clinic every 2 or 3 months or every month, and they need a neuropsych evaluation. Well, we don't do it that often because it's time consuming and it's hard for patients and it's expensive for the neuropsychologist to work. But imagine that in a clinical trial, a patient can go home and they could get data almost every day on their iPhone. Yeah. That expands enormously, reduces the variability in a very highly variable in clinical trials, which is the neuropsychology, which is a primary outcome. So having digital technology is really, that's just one example of speech. We also use a clock people have to draw a clock and put the numbers on it and make it say 10:15 or whatever. And people with early Alzheimer's have difficulty with that. Well, now there's a computerized clock, and instead of getting like 5 or 10 variables out of the clock, we're getting 1,000 variables out of the clock. So it's just the digital technology is just going to change the world.
Howard, one of the problems with you is you're not excited about any of this stuff. So it's just really good. Howard, we have time for one, maybe 2 more questions. So I'm gonna take 2 questions and I'm gonna combine them, but you only have 1 set of time for the answer, okay? So keep yourself honest. There was a question about, these new anti-amyloid drugs and infusions. And the question was about, how long do they need to continue the infusions? And there's another question that says, are we ever going to get to a point where people can take things orally? So can you kind of give us your views on that?
Yeah, those are great questions. Well, I think the really innovative thing about the Lilly trial with donanemab was they had stopping rules. And so that means that, and it makes a lot of sense, right? You do the brain scan at the beginning, or the blood test for that matter, although it wasn't in the trials. And you document a person has beta amyloid in their brain, they have the plaques. And then let's say at 6 months, you repeat the beta amyloid test on the brain, and hopefully one day you repeat the blood test, and it's negative. The drug has done its job. It's just like cancer chemotherapy. For some chemotherapy, people go on it for the rest of their life as a maintenance therapy. But some people, they just get it as a therapy in a trial. They go for a month, let's say, and then the drug is stopped. And I think that's where we're going. We're going to have stopping rules, and that's going to save a lot of money and a lot of burden on patients and their families. An IV infusion over the course of an hour. You have to come into the clinic, and it's expensive and so on. So let's say at 12 patients PET scan at 6 months, or maybe it'll be 12 months. And what Lilly has done, which I think is also innovative, is kind of model, well, if you clear the brain of amyloid, and presumably that has a major impact on progression, how long does it take for the amyloid to start reaccumulating to a point where the disease is back and people need to be treated again? And so you can imagine, you can see this kind of evolving model that sounds a lot like cancer chemotherapy. And I think we're going to have stopping rules, which is going to be really innovative. The other part of your question is something we already have, which is these IV infusions are burdensome, they are expensive, they do work. But the next evolution of this kind of biological therapy, which can't be delivered in a pill, is going to be subcutaneous injection. So, we have EpiPens, we have insulin pens. And I think we've already seen Roche present their data on their subcutaneous trial, which really didn't work very well, but it did demonstrate, I think, proof that this is possible, that with the right dose, with the subcutaneous injection, with a delivery system that's easy to use and patients can take it home, that instead of having to go to an IV infusion center at a major medical center, if you're out in rural Kansas in a town of 122 people, that's not going to work very well. And you get this pen and you can inject yourself. And I think that's not oral. We won't be able to do it orally, but the future of treatment that'll be more tolerable and hopefully just as effective will be these EpiPen type of devices that these monoclonal antibodies will be delivered by.
And what about oral medications? Anything out there funding, anything out there that you see?
Well, there's a lot of oral medications. I mean, a company called Alzion has an oral medication, and a company called Cognition Therapeutics has an oral medication that's going into both of them are in later stage development now. And in different ways, they both try to attack the beta amyloid that's in the brain and remove the plaques. And the Cognition Therapeutics molecule has other pathways on what's called the sigma-2 receptor. The drug started out as an anti-amyloid drug, but seems to have many more downstream effects. And the Alzion drug is actually a new formulation of a drug that I was involved in back in the 90s. That really was a derivative of a supplement called homotaurine, which you find in Red Bull, but it's a new formulation, and it's a real drug, and it seems to. We'll see what the data are. I think it's being developed for people that are ApoE4 positive, and we haven't talked about ApoE4 as the main genetic risk factor for Alzheimer's, but there are, and I'm sure I'm missing many other oral companies that I haven't mentioned.
No, brilliant and terrific. Want to just let folks know that, first of all, anybody who has a question into us, we have your email. We will come back and respond to you directly. So that's number one. Number two, with all that's come out of this summer, we just want to make very clear here, Howard and myself and the ADDF, this year is not going to end quietly. There's going to be a lot more news about Alzheimer's. One, like we've mentioned, Lilly, there's an expectation that before the year is over, that the FDA is going to opine on its approval. And the conventional wisdom at ADDF is that we're looking at a full approval that will happen there. Second, there is in end of October, there's a huge scientific meeting up in Boston, CTAD, called the Clinical Trial Alzheimer's Disease Conference. know that there is a journal, a special edition journal that's coming out parallel to that meeting and will be presented at that meeting. And the title of that journal is The Biology of Aging, Leading the Next Generation of Alzheimer's Drug Development. And what we're really seeing there is that now that we have these anti-amyloids that are breaking through, the field is now moving towards center stage, this biology of aging. Prevention, early diagnosis, prevention, biomarkers, combined therapies, precision medicine that you have. And then after that, we'll hear in the new year about the end of the first quarter, the ADPD meeting in Europe, biology of aging will be a central theme there again. I'm struck by, Howard, that both you and myself and most, if not everyone on this call, as much as we see progress that's going on here, Alzheimer's is something you get drafted to. And I mentioned it before, it's a bit of a societal problem, right? It comes into your family and it just doesn't affect your loved one, it affects the whole family. And we have about a 30-second video clip here. I just want to show of one family, it'd be the ADDF's founding family, the Lauder family, to take a look at if we can pull that up.
I love my mother. And here's a woman who had such light in her eyes and power. She knew something was wrong. My late wife, Evelyn, came in to see her in her home one afternoon and said, "Esty, we miss you in the office." She turned to her and answered her, "I miss me too." What that did for me and for my brother Ronald, it energized us to say, okay, we have to fight this disease and we have to do everything we can to prevent it.
You know, Howard and I wanted to end today with that clip because if you think about it, Alzheimer's comes into your family, and the Lauder family is a family that fought back. And they formed, Howard, with you, the ADDF 25 years ago. And that science that you all had from the beginning is now coming to center stage 25 years later. And it's the power of one family and individuals that can really fight back and make a difference, that move this from a societal problem really to a medical problem, right? That we now have ways to treat. I've mentioned this before, but I'll mention again, the Lauder family recently got together, 3 generations of them, decided to make a $200 million commitment to the ADDF so that we can say to you all, for the next 15 years, every nickel you give us goes directly to the preventions, the biomarkers, the clinical trials that you've heard Howard so eloquently talk about today. Howard has a favorite comment that he's been using in the last year. We are now in harvest time. And the next 5 to 10 to 15 years, the combined therapies, the prevention, the precision medicine now comes to market. So Howard, you're brilliant. The Lauder family is brilliant. And we want to thank each and every one of you that's been on this call today. And we promise you we'll have one more of these before the year is out. So thank you and take care, everybody. Thank you, Howard.
Thank you, everybody.